Hello students. In the last lecture we have studied about the types of canal system in phylum Porifera. Just to have in review, we studied that there are three types of canal system. The first is the Escon type, which is the simplest type. The second type is the Syconoid type, which is little more complex, and the Leuconoid type, which is the most complex. And then there is one more canal system which is found only in larva, and that canal system is called as the Ragon type. Ragon type is found in larva. So today we will study about the body wall. Porifera, they have two layers in the body, two cellular layers in the body. The outer layer which is made up of thin flattened cells called as pinacocytes, and the inner layer which is made up of collared cells having flagella called as the quinoderm. So the cells are quinocytes, and the inner layer is the endodermal layer is called as quinoderm. In between the pinacodum, the outer pinacodum, and the inner quinoderm is present a thick gelatinous layer which is called as mesoglia. And inside the mesoglia, spicules and spongin fibers or both may be found. The cells which traverse throughout the ectoderm and endoderm are the pore cells. Pore cells are present in the body, and the opening of this porocyte or the pore cell is called as the ostia. The plural word for ostia is ostium. So we see that there are two germ layers in the embryo of Porifera. So the embryo, on the basis of embryo germ layers, Poriferans are diploblastic. That is, mesoderm is not present in Porifera. So the ectodermal pinacoderm and the endodermal quinoderm, and in between a non-cellular layer called mesohyl layer is present, which is secreted by ectoderm and endoderm. So we have to remember that mesohyl is a non-cellular layer. It is not made up of cells. The cells, then how does mesohyl arise? So mesohyl is secreted by the ectoderm and endoderm. This is a question which is asked in competitive exam, that mesohyl is secreted by. So it is secreted by both ectoderm and the endoderm. And then some cells from the ectoderm and the endoderm move inside the mesohyl. Those cells are called as amoebocytes. So mesohyl is non-cellular gelatinous layer. The cells which are found in mesohyl, they are not mesodermal. They arise from the endoderm or the endoderm. So they are not having origin from mesoderms. The most important cell which is found in Porifera is the quinocyte, which is found in the inner lining, that is the endodermal lining of the body wall. Quinocyte is a characteristic found only in sponges, and it is also found in a protozoan called as Proterospongia. That's why Proterospongia is considered as a connecting link between protozoa and Porifera. Now this collar cell, if you see, the cell has a collar of processes, and in the center of the collar is present a flagellum. This flagellum helps in forming the water current. So the water is pulled inside the spongiocele by the help of these flagella. If you see this diagram, the colored diagram below, the round body is the quinocyte cell body. Then it has a collar of processes, and then a flagellum is coming. Then you can also see that they help in phagocytosis of food particles. That the food is ingested, food is pulled by the help of flagella in the collar, and then phagocytosis of food is done by quinocyte. So quinocyte is the first cell which will form the food vacuole. So this is question which is asked in exams, that the first cell to ingest food in porifera is, this is quinocyte. So food is ingested, the food vacuole is formed, the lysosomal enzymes are added, and then the food is transferred to amoebocyte where the complete digestion of food occurs. So the first important cell which is found throughout the lining of endoderm is quinocyte, and they are called as collar cell, probably the most distinctive and most important of the sponge cell. Each collar cell has a single flagellum surrounded by sieve-like collar that acts as a strainer, food strainer. Collar is made up of microvilli and microfibrils. The flagellum beats to draw the water currents into the sponge and then to strain particles through the collar. Food is absorbed phagocytosis by the collar cell and then sent to other cells in the sponge. Quinocytes line major activity depends on the canal system. In the whole spongocoel of the ascenoid type, quinocytes are present. But in cyconode type of canal system, the quinocytes are restricted to the radial canals only. They are not found throughout the lining of spongocoel. And flagellated chambers or leuconoid. So quinocytes are restricted only to the flagellated chambers in the leuconoid type of canal system. Collar cells are most identical to the cells of quinoflagellates, and the quinoflagellates are protozoans. That's why quinoflagellates are considered as the connecting link between porifera and protozoa. So the endodermal cells of protozoa are collar cells, which are also called as quinocytes. Pinacocytes, the outer epithelium, that is the ectodermal cells, are the pinacocytes. They are very thin flat cells. Some are contractile, which are called as myocytes in the circular band around oscula to regulate water flow. So myocytes are present around the osculum to regulate the water flow by closing or opening the osculum. Then porocytes. See, these porocytes, they extend throughout from ectoderm to endoderm. So they are tubular cell and they form the pores of ascenoid sponges. That is the ostia. Inner and the outer ostia are formed by the porocytes. Then archaeocytes. These are the amoeboid cells and they are totipotent and they receive food particles from quinocytes for digestion. Then we have sclerocytes, they secrete spicules, spongocytes which secrete spongin fiber, and collinocytes which secrete collagen of the mesohyl. Support. The cells in the gelatinous matrix of the sponge are around the skeleton of spicules. Spicule maintain its shape and keep the pores and canal open. Spicules on the basis of composition are of two types. They are either calcareous or siliceous. The calcareous spicules are made up of calcium carbonate and the siliceous spicules are made up of silica. Spicules often unite to form a rigid network that looks like fiberglass. Example, Venus flower basket. In Venus flower basket, only siliceous spicules are present. So siliceous spicules, they are transparent. That's why the body appears a very transparent and glittering appearance. Spongin, it is a form of collagen, a protein only found in animals. Then it forms flexible protein fibers related to keratin. And spongin is the main part of euspongia, which is called as the commercial sponge. So in commercial euspongia, the whole sponge is taken out from the ocean. It is dried and the spongin fibers are taken out and used as sponge in the furniture industry. So ectoderm, just to revise, ectoderm is spinacoderm, endoderm is made up called as quinoderm. Ectoderm is called as pinacoderm because it is made up of cells called as the pinacocytes, and endoderm is called as quinoderm because it is made up of cells called as quinocytes. There are certain abemocytes in mesohyl and other parts. First is the arcucytes. They are the totipotent cells. Trophocytes, they are called as the nerve cells. And thesocytes, they will store extra food. Gland cells secrete the mucus. Colonocytes secrete the collagen of mesohyl. Scleroblasts secrete spicules. And myocytes, they form the muscular filter around the osculum, and they are modified pinacocytes. Germ cells, ova and sperm, they are found in the mesohyl layer. Chromocytes, again, they store the colored pigments, and phagocytes, which will lead to phagocytosis. Feeding and digestion. See, feeding and digestion, we have to remember that in sponges, no digestion occurs in spongocoel. That is, digestion is intracellular. It occurs inside the cells. All but a few sponges are filter feeders. They feed on detritus, planktons, and bacteria. Pinacocyte, archaeocytes, and quinocytes can all phagocytosize food. Archaeocytes can eat larger particles. Quinocytes can eat smaller particles. Sponges can also absorb dissolved nutrients directly from the water. Digestion is intracellular. This is important. We have to remember that digestion सिर्फ सेल के अंदर होता है. It occurs only inside the cell. So the digestion is intracellular. So no extracellular enzyme for digestion are secreted in porifera. Then some sponges they also act as predator. Cladorhiza corona. It is found in the Mediterranean caves. It lives in stagnant water, not much to filter. So it has developed a tentacle-like appendage covered with velcro-like hooks. The hooks snag shrimp-like crustacea. Within days, other than tentacles, go around victim and engulf and digest it. A small number of sponges are parasites. Boring sponge, which belongs to the class Demospongia. It excavates hollow tubes and passages into the shells and corals, living or the dead host cell or the limestone rock. The animal grows into the canals and holes are created. When boring into live animal shells, the host will either die outright or be much susceptible to predation. May have significant impacts on the coral reefs and oyster reefs, important in recycling shells and corals, that is bioerosion. In some areas, bridge support are no longer constructed of limestone because it is attacked by these sponges. Then no respiratory or sponge organ system. These all the sponges, they directly take oxygen and release CO2 by diffusion. Simple diffusion through the pinacoderm and the quinoderm. A few freshwater forms also have contractile vacuole in quinocytes and archaeocytes for aiding in respiration and excretion. Most important thing that porifera is a multicellular organism, but they are not called as a tissue or an organ. Why? Because the is tissue is not present, and the cells they form a cell aggregate because nervous tissue is absent. The nervous cells are absent. This is very important, that sponge cells can react to local stimuli, but the whole organism will not react to the local stimuli. So no nervous system or no sense organs are present in. So this is about the body wall of sponges. That it is, so we'll just revise The body wall consists of two layers, the ectodermal pinacoderm and the endodermal quinoderm, and in between the ectodermal pinacoderm and the endodermal quinoderm is present a mesohyl layer. Mesohyl layer is a secreted layer which is secreted by ectoderm and endoderm, and some amoebocytes are found in the mesohyl layer, and those amoebocytes are not found by mesohyl. They move from ectoderm and endoderm. That's why the body wall is diploblastic and not triploblastic.